All right, so cell division is a normal part of everything, but it can go wrong in ways that are really scary. So, why is it that it normally works just fine? Big thing is that cell division is very tightly controlled. So, again, we're talking specifically about eukaryotic organisms that are multicellular with organized tissues, because these are the organisms that get cancer, and we're talking about cancer. So different cell types have different behaviors depending on their needs. There are cells that never divide. Your red blood cells, once those are developed, that's it. They're done. They're not going to do any more division. Most of your nerve cells, once they're there, they're not dividing. Your muscle cells, they develop and then they're done. That's it. They're never going to divide again. They're spending all of their time in that phase that we refer to as G0. There are other cells that are dividing constantly. Your skin cells. Skin is subject to a variety of environmental insults, and so it's constantly needing to get repaired, which means a lot of those cells are always dividing, though at a relatively not too excessively fast rate. But cells in the lining of your intestine, those are in such a dangerous, damaging environment. They're constantly dividing really quickly in order to replenish themselves because they're getting damaged constantly. Or we've got liver cells. Your liver cells will sit around in G1 or G0 until there's damage, until something happens, until you take a dose of Tylenol or drink a bit too much or whatever. And then there's damage, and then they're going to divide enough to replace themselves, replace the ones that got killed, and then they stop. Does this actually get controlled? Essentially, there's a combination of go signals and stop signals. The go signals, the growth factors, the things that say divide, 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 these primarily act at G1, at the beginning of the cell cycle, right after mitosis and cytokinesis is finished. The reason that this happens is because you don't want to start making more organelles and dividing your DNA and doing all of that very energy-intensive work that costs lots of nutrients unless it's necessary. And then there are stop signals. There are tumor suppressor proteins, or what they're called. They're called tumor suppressors because when they work correctly, they suppress the formation of cancer. They suppress the formation of tumors. And they're going to say, nope, we don't need to divide. Or maybe we don't have enough nutrients to divide. Or maybe we're going to stop here because DNA synthesis went screwy and the DNA is damaged and we shouldn't go any further until we're finished. Or they cut things off at metaphase because the chromosomes aren't aligned properly. Stuff like that. So normal cell regulation is a combination of both go signals to tell cells you need to divide now and specifically now, and stop signals that don't really do anything unless something goes wrong, at which point they step in and go, nope, we are going no further. You can use an analogy of a car, and maybe we'll even use an analogy of if any of you have taken driving classes, one of those driver training cars, where there's a gas pedal and a brake pedal on the student side, and then the instructor also has a brake pedal that they can use in case stuff gets really out of control. If you don't have gas pedal, you're not going to go anywhere, and you're sort of stuck. But if your brake pedals aren't working, you got a big problem. And if the gas gets stuck down, you got a big problem. And that brings us back to cancer. So this next section, we're going to talk about what cancer is, how it occurs, and how we treat it. So first off, what is cancer? Cancer cells share a certain number of characteristics with them. The biggest one is they divide when they aren't supposed to. Normal cells are going to grow and stop. Cancer cells divide when there's nothing telling them that they ought to divide, or they divide when something should be telling them to stop. So this can cause tumors. Tumors are when you've got a mass of cells in a single place that's causing a problem. And these can be anything from a solid lump of tissue to what we see on the top right there, which is called a teratoma. A teratoma is something that forms specifically from stem cells, and so it will often have multiple tissue types within the same tumor. This particular one has hair on the inside. You can also have teratomas with teeth. They're pretty grotesque, thankfully very rare. Cancer cells forget who they are. Oh, and tumors, you can have one enclosed tumor or you can have something metastatic, which we'll talk about later. Cancer cells forget who they are, what they're supposed to be. They stop doing the job that they were originally differentiated to do. And so you may have a skin cell that starts producing proteins that are associated with a lung cell, or a digestive cell that's no longer actually helping break down food. And they forget where they belong in the body. Most cells, once they have found a place during very early development, during that fetal development stage, that's where they are. That's where they stay. They have no need to move. They have no mechanism to move. They'll just be there. But cancer cells may decide to go wandering. And that's what we call metastasis. And that is where you get some really nasty cancers. And finally, cancer cells become immortal. They stop aging. They stop dying. Whoops. Sorry about that. Did it again. This handsome couple in the lower right here, this is Henrietta Lacks and her husband. So Henrietta Lacks is a black woman who had a nasty cervical tumor. She was in the right/wrong place, the wrong time. She had a very nasty and extremely invasive cancer of the cervix. She went in to the doctor to see about it. The doctor ended up taking a sample. And this was back in the days when people were trying to figure out how we could culture human cells outside of the body. And one of the things that people ran into was the fact that the cells would live for a while and divide for a while, and then they'd all just sort of die. They only had so many divisions in them before they just, frankly, tired out and stopped dividing. And so her cervical tumor was cultured in a dish, and it didn't die. It kept living. In fact, descendants of Henrietta Lacks's cervical tumor are still alive in labs all over the world today. She passed rather shortly after this particular encounter with a doctor because of her cancer. It again was extremely aggressive. But her cells, some of her cells, the deranged cancer cells, live on, completely immortal, capable of never dying. It's interesting and it's a bit horrifying. And if you want to learn more about this, I would strongly recommend the book The Immortal Life of Henrietta Lacks by Rebecca Skloot. It's a really interesting dive into both the biology and the ethics of what went on with this case, which is, at least to me, fascinating, but also really horrifying.